When most people hear the word microbe, they think of germs and infection. But that is a tiny slice of the story. A microbiome is not just a list of microscopic organisms sitting passively in one place. It is a living community in motion, a network of relationships, chemical signals, competition, cooperation, and adaptation. In other words, the microbiome is better understood as an active system than a static inventory. This matters because life on Earth, from humans to crops to marine animals, does not function in isolation. It functions through partnerships with microbial life. The concept itself combines two ideas: small life forms and the larger environment where they interact. So a microbiome includes organisms, but also context, including physical conditions and biological dynamics. Temperature, nutrients, pH, oxygen, moisture, and neighboring species all shape what that community becomes and what it can do. If you remove the context, you miss the logic. That is why modern science treats the microbiome as both who is there and what they are doing together. A key distinction helps clear up confusion right away: microbiota versus microbiome. Microbiota refers to the living members, the actual organisms present. Think bacteria, archaea, fungi, algae, and small protists. Microbiome is broader. It includes those organisms, their genes, their metabolites, their interactions, and the environmental stage where all this happens. Some researchers also debate whether phages, viruses, plasmids, and mobile genetic elements should always be counted as core members. That debate itself reveals something important: this field is still refining boundaries as evidence grows. But the practical takeaway is clear. If microbiota is the cast, microbiome is the full production, including script, set, chemistry, and behavior. That framing avoids a common mistake, where people reduce microbiome science to simple percentages of microbes without understanding functional consequences. Two communities might look similar in broad taxonomy but behave very differently if their chemistry and interaction patterns differ. One of the most powerful parts of the microbiome idea is what some scientists call the theater of activity. Microbes are constantly producing and responding to molecules. These molecules can inhibit competitors, recruit partners, alter nutrient flow, and shape survival under stress. Secondary metabolites are a major part of this hidden economy. They are not just side products. They can define who thrives, who declines, and how stable a community remains under pressure. Another crucial process is quorum sensing, where microbes release small signaling compounds and detect population density. Once a signal threshold is reached, group behaviors switch on. That can include adhesion to surfaces, coordinated enzyme production, collective defense, or biofilm formation. Biofilms are especially important because they turn scattered cells into structured communities with shared protection and resource gradients. So the microbiome is not only about presence, but also coordinated decision-making. These systems display a kind of distributed intelligence without a central controller. The microbiome is not unique to humans. It is universal across plants, animals, and many habitats on Earth. Every host is an ecosystem partner, and every habitat has microbial architecture. In marine science, researchers once focused mainly on one host, one symbiont stories, because those systems were easier to isolate and describe. But that approach underestimates real-world complexity. Marine animals host diverse, multi-member microbiomes, and those communities can influence physiology, immunity-like defenses, behavior patterns, and ecological role. This shift from pairwise to community-level thinking is changing how scientists interpret adaptation in the ocean, especially under climate-related and human-induced stress. If temperature shifts, acidification, pollution, or nutrient disturbance changes the microbiome, host performance can change too. That means adaptation may depend not only on host genetics but also on microbial flexibility. In this view, resilience is shared. The host and microbes co-produce the ability to tolerate change. Plant microbiomes show the same principle with direct implications for food systems. Plants carry microbial consortia inside tissues, called the endosphere, and on external surfaces, called the episphere. These communities are not accidental passengers. They help regulate nutrient uptake, stress tolerance, defense against pathogens, and growth performance. Researchers often discuss a core plant microbiome that includes keystone taxa, organisms whose influence is disproportionately large relative to abundance. Remove a keystone interaction and the whole system can destabilize. Keep it functional and plant fitness improves. This perspective changes agricultural strategy. Instead of only asking how to kill harmful organisms, science now also asks how to maintain beneficial microbial structure across soil, roots, leaves, and surrounding environments. The plant becomes part of a holobiont, a functional unit composed of host plus associated microorganisms. In practical terms, crop performance and ecosystem health depend on preserving relationships, not just managing individual species. In mammals, the gut microbiome has become a central model for host-microbe interaction. The key point is not that microbes influence one narrow pathway. It is that they participate in broad physiological regulation, including metabolism, signaling, and immune calibration. Over evolutionary time, host lineages and microbial lineages have co-evolved. That means adaptation did not happen on one side alone. Mammals diversified into different diets, habitats, and lifestyles while microbial partners also shifted. This coevolution helps explain why microbial communities can be deeply linked to host function, yet still vary across species and environments. It also helps explain why simplistic one-size-fits-all narratives about the gut are often misleading. Functional outcomes depend on ecology, history, and context, not just on a single microbe labeled good or bad. Understanding the gut microbiome therefore requires systems thinking, network interactions, functional pathways, and dynamic balance over time rather than static snapshots. Historically, microbiome science did not begin with modern sequencing. Its roots trace back to early microbiology in the 17th century, when microscopes first revealed an invisible world. At that stage, disease understandably dominated attention because infectious causes were urgent and observable. But as methods improved, a major correction emerged. Only a minority of microorganisms are primarily associated with disease. Most microbes are either neutral in many contexts or actively beneficial for ecosystem function and organismal health. That reframing was not a minor update. It was a paradigm shift from pathogen-centered thinking to ecology-centered thinking. Microbes moved from villains at the edge of biology to foundational players in the center of it. The story of microbiome science is therefore also a story about scientific perspective. What you can see shapes what you believe, and what tools reveal can overturn decades of narrow assumptions. The modern acceleration came from molecular and cultivation-independent techniques. DNA discovery, PCR, cloning, and then high-throughput sequencing made it possible to study communities without growing each organism in isolation. That changed everything, because many microbes are difficult or impossible to culture with standard lab methods. Once metagenomic approaches matured, researchers could analyze genomes and community-level genetic material directly from natural habitats. Suddenly, microbial ubiquity was measurable at scale, and functional potential became traceable through genes and pathways. This did not just add detail. It transformed the questions scientists could ask. Instead of asking which single species causes one effect, researchers could ask how communities assemble, how functions are distributed, how interaction networks reorganize under stress, and how host-associated microbiomes differ across development, geography, and environmental conditions. The result is a richer but more demanding science where interpretation requires integrating ecology, evolution, chemistry, and computational analysis. A crucial insight from all this work is that microbiomes are dynamic, not fixed signatures. Community composition can shift with diet, season, developmental stage, stress exposure, habitat disturbance, and host condition. Functional output can shift even when high-level taxonomy appears stable. This is why definitions now emphasize dynamism and why updated expert frameworks distinguish clearly between microbiota and microbiome. If we ignore change over time, we risk mistaking temporary states for stable rules. If we ignore function, we risk overinterpreting who is present while missing what is happening. Dynamic framing also improves interpretation of interventions. Any attempt to modify a microbiome, in medicine, agriculture, or conservation, enters an adaptive system that can compensate, resist, or reorganize. That means expectations should be realistic, outcomes are probabilistic and context-dependent. Good microbiome science therefore combines longitudinal observation, mechanistic experiments, and careful ecological reasoning instead of quick static conclusions. So why does this topic matter beyond specialist labs? Because microbiome thinking changes how we define health, productivity, and resilience across domains. In human biology, it encourages system-level approaches rather than single-cause reductionism. In agriculture, it supports strategies that protect beneficial microbial relationships crucial to plant performance. In marine ecology, it highlights how climate stress may act through host-microbe partnerships, altering adaptation potential and ecosystem stability. And across all fields, it reminds us that life is relational. Organisms are not sealed units but participants in layered biological networks. The microbiome concept captures that reality with unusual clarity: communities in context, structured by interaction, responsive to change, and essential to function. If there is one idea to carry forward, it is this: to understand any living system, we must understand the microbial worlds entwined with it. The hidden majority is not background noise. It is part of the engine. If you found this helpful, please like this video and subscribe for more explainers.